recording. Okay. So hello everyone, welcome to the Mineral Life seminar. So I'm happy to introduce Dr. Clara Chan, Associate Professor at the University of Delaware in the USA. So a few words to present her. So she completed her PhD in 2006 in UC Berkeley on Earth and Planetary Science. After that, she became a postdoc at the University of Southern California in marine chemistry and geochemistry. And she joined the University of Delaware in 2009, where she is now working on the mineral life interaction, and more specifically on oxidizing, on iron-oxidizing bacteria. So today, she will present her multidisciplinary approaches on how iron-oxidizing bacteria thrive and drive a geochemical cycle. So we are really excited for this talk. And as usual, the seminar will last for about an hour with some time for the question at the end. So you can also write your question in the chat and we will read them out loud at the end of the seminar. And so that's all for me. The floor is yours. Thank you.
Great. Thank you so much for having me here. I'm very excited to talk with this group that works at the interface of bio and geo and minerals. Can you see the right screen?
Yes, we can.
Okay, let me just adjust my own. Okay. All right, so I like to start by acknowledging all the people who really did the work, which are my students, former student Sean McCallister, postdoc Jessica Keffer, former student Rebecca Vanzura, and my student who's about to graduate, Nanqing Zhou, as well as a number of different great collaborators and our funding sources. So I have been studying iron since I feel like the beginning of time. And this is why there is just so much of it on Earth. We literally live on a hunk of iron. And iron is so central to the environmental cycles and that drive so many of the things we care about, right? Water quality, soil quality, etc. And so much of this is because of the redox activity of iron. Iron is also essential to a lot of the materials that are useful to us: steel, catalysts, sorbents, etc. Microbes can take advantage of this vast iron source and both oxidize and reduce, but we're going to focus on iron oxidizing microbes. And within them, there are many different kinds, but our focus is on ones that oxidize iron 2 to iron 3 and couple that to breathing oxygen as we do. So I will call these Feob, iron oxidizing bacteria. And it's not an easy life. This is a high potential metabolism, high redox potential, and so there's not a lot of energy. So they need to go through a lot of iron to survive. There's competition with abiotic iron oxidation. And then they make mineral products, which of course we love as mineralogists, but is a challenge for these organisms because they have to do something with them so they don't get encrusted. Geochemists often ask me, well, you know, why does it matter if it's microbial? Iron is just going to oxidize, rust will happen. But the importance is that organisms connect iron cycling with nutrient cycling, carbon and nitrogen cycling. And so if we are to understand their ecological and biogeochemical roles, we need to know how this happens. And so we have to look inside the microbes. So since we are in Zoom, I want to briefly bring you to Delaware, not just my office, but this is across the street from our campus. And these are iron seeps that are flowing into the Christina Creek. And so these iron seeps are everywhere where you have humid, wet environments. So there are a dime a dozen here. And this is actually Dave Emerson, who was quite influential in my career. And he is comparing his orange shirt to the very orange and fluffy iron microbial mats. So this is one of the more obvious expressions of microbial iron oxidation in the environment. Maybe you have seen it. But we see iron oxidizers all over in maybe more subtle ways, from the deep sea, which is not so subtle, to coastal environments, weathering rocks, in sediments, in groundwater. Wherever we look and we see iron oxides, we're able to find iron oxidizers of some sort. And so they are widespread. Specifically, neutrophilic aerobic iron oxidizers grow where anoxic fluids meet oxic environments. And so aside from the seep, they are environments like the rhizosphere, where you have maybe a wetland plant growing in reduced waters, but the roots bring oxygen down into the sediment. Or there's a coastal groundwater discharge where anoxic coastal groundwater flows out into the ocean. In groundwater wells, where we poke a hole into the anoxic aquifer, and these organisms love to clog the wells, which is a problem. In all of these environments, these organisms make biominerals, and these biominerals, these iron oxyhydroxides, are very sticky, and they will absorb phosphate, organic carbon, a whole host of different metals. And so with iron cycling goes many, many other cycles. And so in this other way, iron is central to our biogeochemical cycles. So for these reasons, we want to be able to detect iron oxidizers in the environment and be able to understand their roles and quantify their contributions. But there are many, many challenges to this. And from the biological side, right, we, up until recently, didn't know the genes involved, or they're poorly characterized, right? So we couldn't use genes like we do for nitrogen fixation and carbon fixation and sulfate reduction. We just didn't have that ability. This is directly connected to the fact that they're difficult to grow, and there's low yield, and there's no genetics in these systems. And so it's difficult to apply classical biological techniques. And on top of that, as you may already know, minerals make a lot of extractions and other biological techniques difficult. So everything is a challenge in working with these organisms, but we persist. And so one of the central questions for our recent work over the last number of years is, what are the iron oxidation genes? Because if we can understand the genes, then we can use the power of omics to understand their contributions in the environment and perhaps even quantify. And the reason is because there are not other markers, and there is so much abiotic oxidation, right? We really need a marker. And so we took the approach of transcriptomics and meta-transcriptomics and genomics combined with functional biochemical assays to try to find and validate this gene. And so for the rest of the talk, I'm going to first, I have to show you the minerals first. So I'll show you the minerals and the mass first and introduce you to our major field site in Hawaii. And then part two will be using metagenomics and metatranscriptomics to narrow down the iron oxidation genes, and then a biochemical characterization, and finally applying what we know to detecting new iron oxidizers and understanding how they drive carbon and nitrogen cycles. All right, so let's go to Hawaii and look at the biominerals. So these aerobic iron oxidizers are famous for making pretty minerals. And I just want to point out that it's not just one group, right? It's this dichotomy of the beta proteobacteria and the zeta proteobacteria, this very strange coincidence, but perhaps not a coincidence, that these organisms make the same twisted stalk, and then there are at least two different organisms that make the iron encrusted sheaths. And so the inference is that this is an adaptation to the iron oxidizing metabolism, one way of dealing with all the iron oxides that these organisms make. And so our model organism in this case is the zeta proteobacteria, Mariprofundus ferrooxidans, PV1. It is an isolate from the Hawaii site that I'll tell you about. And here the cell, over the course of, this is about an hour and a half, is oxidizing iron 2 and packaging it in this mineral and organic based stock. And over time, as it oxidizes more, it makes more stock and the cell moves and it twirls. This is a cryo TM tomography reconstruction. And you can see the cell is very clear. There are no minerals in the cell. There are no minerals on the cell. This is a very clever cell, able to package its waste very specifically into, this is the beginning of the stalk right here. And so this is really quite an amazing feat and a powerful adaptation. What's also cool is that when the cells divide, so down here you'll see, as the cells divide, the stalk branches. And so this can be seen actually in the fossil record, right? And so this is an immediate record of the fundamental characteristic of life, which is reproduction, which is also very cool. And I will change my slide. Okay, so let's go to Hawaii. Our field site is a submarine volcano off the coast of the big island of Hawaii. So the peak of the volcano is about 1 kilometer below sea level. And although it's not erupting now, there's still active hydrothermal activity throughout the volcano. When we go down, or more precisely, when we send the robot Jason down, we see a lot of orange. This is really one of the nicest places in the world to be an iron oxidizer. You can see the shimmering fluids. These are about 50 degrees C fluids, maybe 60. And everything you see here, you'll see it jiggle. This is iron microbial mat. So these organisms are taking advantage of these iron-rich fluids, dissolving iron from the basalt. And it's within an oxygen minimum zone, you'll see in the next slide. And so they live at this interface of oxygen and iron and create these really lush, bright orange microbial mats, all made of biomineral. So we, more precisely Brian Glazer, took a profile through the mat. And you can see right at the mat surface is where you see the consumption of iron and oxygen. So that's representative of the metabolism. And at the interface, the oxygen is at micromolar levels. And these are the microaerophiles adapted to living at low oxygen levels where they can compete with abiotic oxidation. We take pieces, so pieces from that device that you just saw, and embed them and section them to look at the structure of the mats. And this is how we know that they're completely made of these individual twisted stalks. That creates the vast, large mats that we see, these microstructures. And so it's amazing, these tiny micron-sized organisms are making centimeter scale and then meter scale mats. And you can see individual colonies. They're a bit flattened because they got flattened in the petri dish, but you can see the branching structures as well. So that accounts for these colonies. And so colony upon colony upon colony is how we build these large structures that become these extensive mats that we see when we go visit the seamount. And so these microbes are using autotrophic iron oxidation to build the physical framework of the community, but it's also building a chemical framework as well which we'll talk about later. And so how are they doing this? And I do want to say that not every iron oxidizer makes a structure like this, but we're using this as a model system because we can really tell that it's biotic. But when we go to aquifers and sediments, we don't necessarily see these things. And so it's not that we can use biominerals every time to detect biotic activity. We need the genes. And so here we know it's biotic. And so we're looking for the genes in this model system in order to be able to apply them to other systems. Okay, so how do we do this? We started out by just imagining maybe every iron oxidizer has the same gene. Could we be so lucky? And so we use comparative genomics of the handful of isolates that existed. So this is a crude Venn diagram, but just to say, there are phototrophic and acidophilic iron oxidizers. There are neutrophilic iron oxidizers, which I've broken down into the marine and the freshwater ones. And now we not only have isolate genomes, but we also have hundreds of metagenome assembled genomes as well, right? So this is kind of an iterative process, but originally this was mostly with isolates. And the question is there one gene in common? This was harder than it looked, right? But the answer was amazingly yes. There was one gene, and this is the amino acid tree that goes along with it, that is a cytochrome gene. And it was originally characterized in AcidothioBacillus ferrooxidans and named CYC2, so cytochrome C2. We call it Cyt2. And it was functionally characterized in AcidothioBacillus. I will note that the branches on this tree are very, very long. And part of the difficulty was that back before we had HMMs, we had to blast and then blast again, meaning we had to walk the blast because you can see how long these branches are. And so it turned out there was another gene, another protein that was functionally verified. And it turned out to be one that I worked on in my PhD, but I just didn't know it because it was so far away and it took a while to find it. But this is a cytochrome 572 and another acid mine drainage iron oxidizer, leptospirillum. But you see there are many, 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 many branches on this tree, meaning it is actually quite a common gene. And the question is, are these all iron oxidases? So that's a hard question to answer. But you can see that the neutrophilic iron oxidizers, the Gallianella, these freshwater iron oxidizers, and the zeta proteobacteria form a relatively tight cluster over here, and we call it cluster 1 because it's the one that we want to investigate. And also clusters with these phototrophic neutrophilic iron oxidizers. And so because it's so far away from these functionally verified ones, we wanted to find out, do these oxidize iron? Because if they do, then we can use this gene to detect iron oxidation activity. And also there are other branches here, right, to determine if these are also putative iron oxidizers. Okay, so normally you would do this in an isolate, and we will come to that later. But at the time, it was more practical to send a robot to the bottom of the ocean and perform genome-resolved metagenomics and metatranscriptomics to look for the expression of this and other genes and to understand how this gene expression responded to iron oxidation conditions. And so we took samples, we assembled genomes from those samples, and then we mapped the transcriptomes to individual genomes. And then we also performed these incubation experiments on the ship where we added iron and looked at the response of the genes to the addition of iron. And that's how we connected function to gene expression. So this is the PhD work of Sean McCallister. And he loved to go on cruises. And so I will mostly tell you about Hawaii, but and also some of his results from the Marianas, but he went on 3 cruises. And so you can imagine his PhD took a very long time. from these many samples, we are able to get a comprehensive library of zeta proteobacteria genomes. And I joke that he should have labeled his dissertation, everything you wanted to know about zeta proteobacteria, or maybe everything you wanted to know and things you didn't want to know. But what you're looking at here is a concatenated ribosomal protein tree from all of those genomes. And so the zeta proteobacteria, I'm happy to say, is still a class of proteobacteria, but it's really, the diversity is about order level. And there are two families, it's family 1 and family 2, and comprising about 11 genera, a number of OTUs and species. We were so obsessed with zeta proteobacteria that at the time I got a new dog and I also named her Zeta because apparently I couldn't think of another name. Anyway, the isolates of the zeta proteobacteria are mostly around Mary profundus, and Dave Emerson has a few Georgia isolates, but for the most part, the zeta proteobacteria are uncultured. And so the question is, are they all iron oxidizers? And do they all express the iron oxidation pathway? It's a bit of circular logic, so please bear with me while we go through this story. And so you see this tree again, and it's color-coded by the different zeta taxonomic units. And those individual taxonomic units have multiple genomes. And so we looked for our hypothesized iron oxidation pathway, including the CYC2 gene, which is an outer membrane cytochrome gene, and asked, do all members of the zeta proteobacteria have it? And the answer is yes. And which is remarkable, actually, because when we look for the rest of the iron oxidation pathway, we see it's a mix and match, but they all have this particular gene. OTU2 is a bit hard to assemble, so it's a bit of an outlier. Not only do they all have this putative iron oxidation gene, when we looked at the full pan-genome, so now every genome is a line here, you can barely see it, and then every gene is a column. When we look for other metabolisms besides iron oxidation, we hardly find anything. It's very rare, unless of course we don't recognize them, but there are a lot of metabolisms that we should recognize, right? Sulfur oxidation, etc. Organic oxidation, we don't see those. And so this emptiness suggests that zeta proteobacteria are actually an entire class specializing in iron oxidation, which is consistent with where we find them. We tend to find them in association with iron. That also implies that iron oxidation requires a lot of adaptation, right? That these organisms have the corner on the market. They're able to oxidize iron and deal with all the challenges that come with that. All right, so they have the gene. What happens next? So because we do genome resolved transcriptomics, we're able to see the level of expression of the CYC2 gene. And so we would posit that if it's important in this iron oxidizing environment, in these hydrothermal vents where there's basically nothing else to eat, that it should be fairly highly expressed. So what I'm showing here is how high it's expressed. And we show this in percentile, right? So 100 percentile is the top expressed gene. And for the most part, so the average is 93 percentile, which is quite high. But you see for many of these organisms in all three sites, right? It is the top or near the top expressed gene. And so it's very important to these metabolisms. The ones that are exceptions tend to be the deeper branching, the family 2. Okay, so they all have the gene and they're expressing it highly, but are they expressing it in response to iron oxidation? And this is where the shipboard incubations come into play. So we put the mat in a serum bottle, we control the oxygen in the headspace, and then we add iron. The mat has been sitting on the ship for a while, so it's all out of reduced iron, right? It needs some electron donor. So we add that and we document the oxidation of iron, meaning in here the iron 2 is going away. And we document that that is biotic by azide killing the mat and showing that it's much slower. So we have biotic iron oxidation activity. And then we track the expression of this gene that you now know and love over time, mapped to specific genomes. So this is one particular, this is the most abundant zeta proteobacteria in this sample. And we can see after we add the iron, the gene expression goes up. And so we can see the direct response of gene expression to the addition of iron or the creation of iron oxidation conditions. This is true overall, although it's a subtle effect and it differs by the organism. But overall, from our meta-omics, we see that in this very highly iron oxidizing environment, Cyc2 expression corresponds to iron oxidation. We chose that word carefully. So thus far, we have mounting evidence that it is an iron oxidation gene. And so we turn to biochemistry to try to actually prove that the protein oxidizes iron. And this proved to be an extremely, extremely challenging project, which I'm glad it was finally successful, but it took several years of effort from my postdoc, Jessica Keffer. What we did was to take the gene from Mary Profundus and put it in E. coli and ask E. coli to express it. E. coli did not want to express it, but we made it do it anyway. Many trials and tribulations later, including trying to submit a manuscript without having done this and getting rejected, we did express it well enough to purify it, not to purity, but to the extent that it was the only cytochrome in the eluent. Then we could use the heme UV-vis spectra to monitor activity and assay the actual protein for iron oxidation. This is the UV-vis spectra. It has a classic heme spectrum. And so when we add iron to this enriched or purified CYC2 that was expressed by E. coli, we can see a shift, and these shifts represent the electron transfer from iron 2 to Cyc2. And so this is the proof that the protein actually oxidizes iron. Now, cytochromes can be promiscuous in the sense that maybe it oxidizes iron in vitro, but it was not necessarily built to do this in the organism. And so we went further and checked the redox potential, because we would theorize that if it was built to oxidize iron in the system, it should have a particular redox potential. And so here's a redox ladder, here's 0. And the iron 2, iron 3 couple has a redox potential of 0 plus or minus 100 if the product is very hydrate, which it is. And so in order to conduct downhill electron transfer, right, the redox potential has to be below this, which it is. The redox potential is about 200. But it can't be too low because these organisms are autotrophs and they must conduct reverse electron transfer in order to make reducing equivalents for biosynthesis, in particular, the fixation of carbon, right? And so I say that this redox potential is just right. It is below iron 2, iron 3, but is not so low that this is a difficult task, because this requires energy. And so we finally show that this cytochrome, CYC2, actually oxidizes iron. And so we can much more confidently say that it is an iron oxidase. Our story doesn't end here, because it is not the only posited iron oxidase, which is good because, you know, there's a lot of iron in this world. There's probably a lot of different ways to oxidize iron, but this one is the one that we think is most universal. And the question is why? So the other iron oxidases that are posited are multi-heme cytochromes. And so here in this cartoon, the heme groups are boxes, black boxes, and their outer membrane, right? And so there is a porin that houses the cytochrome in the outer membrane. Cyc2 is a fused, it's one protein, right? It's one CXXCH heme binding group fused to a beta barrel porin. It is relatively small. It's the normal size of a porin. These other multi-heme outer membrane cytochromes, which you may know from the Chiwanella, the metal reduction, MTR, CAB. These are much larger and would require more investment by the cell. There is a posited iron oxidase. Actually, there's a known iron oxidase, the PIO-AB system from Rhodopseudomonas ferroxidans. That has been genetically verified. But there are these MTO-AB homologs that are thought to be iron oxidases. Why would you have this? Why would you invest that much energy into a larger, more complex protein? The answer may be that this spans the membrane, whereas this doesn't, right? One heme is not enough to get an electron across the membrane. And iron would have to go into this porin and be oxidized, and then the iron-3 would have to be shuttled out. In this case, this could actually contact something on the outside, perhaps a mineral. And so our hypothesis is that Cyc2 is for dissolved iron oxidation, and these other ones may allow organisms to oxidize minerals. Okay, and so finally, we reached the point where we need to test this pathway in an isolate. I told you they are challenging to grow, and so that's why we didn't start with them in the first place, right? There are a lot of problems that we have to solve along the way. But we're lucky to have an isolate, Sideroxidans lithotrophicus. It's a relative of the Gallionella. It does not make any fancy minerals. It just makes granular minerals. But its superpower is that it can grow on a number of different substrates, including both dissolved iron, thiosulfate, it has various sulfur oxidation genes, and then also solid iron, including this iron clay, iron-2 smectite. And in its genome, it has the two different iron oxidation genes or gene clusters, the Cyc2 and MTO. And so we can ask the question, what does it use for what? We started with dissolved iron, frankly, because it's easier. We actually use iron chelated to citrate. And so these are the putative pathways, right? The Cyc2 and the MTO. And we looked at the gene expression and we normalized that to a housekeeping gene, JRB. It turns out that apparently Cyc2 is so important to sideroxidans, it has 3 copies in its genome, all 3 in a row. And what we see is the expression level of all 3, but particularly the first one, is quite high relative to the MTOA, which is this one, right? And so on dissolved iron, it looks like the CYC2 is actually one of the top expressed genes. And it looks like that's the gene that corresponds to dissolved iron oxidation. Because we look at the full transcriptome, right? We can look at the entire pathway. And in fact, we can refine the pathway based on what we found when we compare the expression of cells grown on iron-2 versus the expression of cells grown on thiosulfate, right? So this is a differential expression. And so these arrows tell you which genes are upregulated highly or less highly or downregulated on iron when compared to thiosulfate. This one, the Cyc2 one that's very highly expressed, is so highly expressed it kind of doesn't matter. I think it's almost constitutive. So it's always on and always high. But these other 2 copies go up on iron. And we think that's because, remember, there's not a lot of energy from iron. And so if you need to oxidize a lot of iron, you probably need to make a lot of iron oxidation proteins. And so that's what the additional copies probably are for. Oh, there's iron out there. I'm going to try to express more Cyc2 proteins. We also see the high upregulation of the reverse electron transport and carbon fixation. And so we see that iron oxidation drives carbon fixation according to the transcriptome. And that's a direct link. We think of the main electron sink as oxygen, but in reality, CO2 is an important electron sink here as well. And so one of the main outcomes from this work is that we not only get to understand what is the iron oxidation gene, but we get the full pathway, which actually is really useful if we want to detect activity in the environment, because we can see not only the iron oxidation activity, but the consequence for physiology and metabolism in the cell and the consequence for carbon metabolism. So this is our summary cartoon, but ultimately it means that we validated and we fleshed out this pathway. And so we can use multiple genes actually to look at activity. We haven't made such a nice cartoon for our mineral oxidation experiments yet, but I will just tell you here that we see increased expression of the multi-heme cytochrome gene when we grow these organisms on solids. We actually see the expression of both Psych2 and MTOA. And that's a bit of a longer story than I'm happy to talk about in the question period. But we really only see the expression of MTOA when we add this mineral. And so we think that that is the purpose that our hypothesis thus far is proven correct, that Psych2 is an oxidase of dissolved iron 2, which is quite common in the environment. But as we know, as earth scientists, minerals are where most of the iron is. And so if organisms have a way of gaining energy from mineral iron, that's even better. And so I think that they have these more complex multi-heme cytochromes in order to do that. And ultimately, either way, this iron oxidation fuels carbon fixation, and CO2 is actually an important sink, electron sink for these organisms. So, okay, I told you at the beginning that we care about these organisms, not just how they're oxidizing iron, but how they contribute to nutrient cycling in the environment. And so I just want to finally say, how we do this. And we're going to have to go back to Hawaii to understand this. And so I'll just, I know that there's not a ton of time left, so I'm going to quickly tell you about how we do it by showing you this video where we sample the top of the mat. So remember, the organisms are making the mat. They're leaving the stalks behind, right? The cells are mostly at the top of the mat. That was the delicate sampling. This is the non-delicate sampling. We call the slurp sampler, and then the scoop sampler, where we unceremoniously vacuum up or scoop up as much of the mat as possible. And so we actually get deeper parts of the mat. And so these represent different communities. The top, which is this syringe sample, S1, and then the bulk community, which is mostly the lower community. And so we can see this. The surface mat is mostly zeta proteobacteria, which corresponds very nicely with our model of mat formation. And these bulk mats have much more diversity. There are many organisms living in this apartment building of the framework of stalks. And in particular, this purple one, I want to call your attention to, which is a novel organism in the DTB120 phylum that we had to name because there was no closely related organism, and we named it Fairystratum. And we have no idea at this point what it's doing there, but it's apparently very happy in these iron-rich mats. Using genome-resolved metagenomics and metatranscriptomics, we can reconstruct biogeochemical cycles. And I'm just going to walk you through them, but this is just a summary of who is doing what, right? We can tell who is doing what. Who is oxidizing iron? Well, no surprise, the zeta proteobacteria. You could have guessed that as well as anybody here. But what we were surprised about is when we look at the bulk samples and we look at who is oxidizing iron in the deeper mat, we use our knowledge of Psych2, right? We now understand that this is an iron oxidation gene. And then we find it in this novel organism, Fairystratum. And how do we know it's an iron oxidation gene? Well, we have to use our tools here. We put it in the tree, and it turns out that the site 2 is in this cluster 1 within all of these neutrophilic iron oxidizers. So we infer its function due to its homology. So far, so good, but we'll come back to that. Who is fixing carbon? Almost all the carbon fixation, according to the transcriptome, is happening at the mat surface. That's because it's coupled to iron oxidation, right? And it's happening in the zeta proteobacteria. And so these organisms are fueling primary production, which then can be used and is the chemical foundation for this community. Nitrogen is always more complicated. And so there are many organisms cycling nitrogen, but I want you to call your attention to, again, the purple organism, the fairy-stratum, that is expressing NAR-GH. This organism is a nitrate reducer. Perhaps it's coupling iron oxidation with nitrate reduction in these deeper mats where there is no oxygen. And so we go back to our incubation experiment, which is our only tool for assessing activity in these mixed community systems. But we use genome-resolved transcriptomics in order to look at the activity of particular organisms. And this time we're looking at the fairy-stratum. And when we add iron and we look at the gene response, we see the coincidence increase in both the nitrate reduction gene and the iron oxidation gene. And so this suggests that the activity of iron, this DTB120 fairy-stratum, is the nitrate reduction coupled to the iron oxidation, which is the best we can do without trying to isolate. And so altogether, we can make cartoons in which we see that there are many different ways in which iron oxidation is connected to nitrogen cycling. But it all starts with these primary producers, the zeta proteobacteria, and then now we can put the purple organism, fairy-stratum, in this picture and see how together they drive carbon and nitrogen cycling. And you can read more about it in Sean's ISME paper. There's a bit of a virus story as well. So much of what we've talked about is basic science, but I just very quickly want to say that these are very useful organisms, right? You probably already know that they can be used in bioremediation. We have projects now to try to use them to recover metals from mine drainage systems, particularly these rare earth elements that we really need. And these mats, if you look at them, just the pictures, they remind us of fabrics, right? But those are materials. And the question is whether or not we can harness this to make useful materials. But in all, what we know now is that they not only make beautiful minerals, but they have evolved these outer membrane cytochromes, multiple of them, even more than the ones that I've talked to you about, I think, to be able to oxidize iron, keep it outside the cell, and create these structures. They're widespread, and through their metabolisms and through the mineral reactivity, these organisms drive carbon, nitrogen, and metal cyclings in a whole wide variety of ecosystems. And so that's the end, and I am happy to take questions as there is time.
Thank you very much for this talk. It was really thrilling. So if you have a question, please raise your hand or type it in the chat and we will read it. I actually have one of my own. I was wondering, so in the really beginning of the talk, you were describing gamma proteobacteria, I guess, that were forming tubes of iron mineral. And I was wondering if you also find those cytochrome inside those kind of bacteria.
Yes. Okay, if I could use my PowerPoint a little bit better, just for inspiration. Right, so the sheath-forming organisms, the leptothrix and the zetathrix.
Yes.
So funny you ask, we have started working with Leptothrix chladnii, which is known as a manganese oxidizer, but it is in culture. And it turns out that it has many cytochromes as well as multi-copper oxidases. It's a little bit different, right? Because the cells sit inside the sheath. And so it's actually even more complicated situation. But when I looked at the genome, it has an almost 5 megabase genome. There are just so many options. And so there's probably many different mechanisms for many different situations to oxidize both iron and manganese and maybe other metals.
Okay. So we have to invite you again in a couple of years then. So Geoffroy had a question?
Yeah, just still on this slide actually. How does the zeta proteobacteria do to orient the biomineralization? Because as you told us, the zeta proteobacteria is able to produce its biominerals only in one direction. How does it do that?
Oh, I would love to know. So there are a number of interesting biophysical, and this is really going to be a rhetorical answer, biophysical and chemotaxis questions here, right? So biophysical, one, how does it even know which side of the cell, right? It comes out of the curved side of the cell. I mean, how does it even make that shape and how does it know? This is part of what I think is, taking up a lot of its genome and causing the adaptation. It's a very complex thing that it has to do to know how to put those minerals there. And then it has to know, okay, I am putting the minerals on that side of the cell. That means I am going to move this way, right? So it has to orient itself in the gradient to make sure it moves in a particular way. And I actually met with somebody who studies motility, and he agreed that actually the formation of the stalk or the sheath is a form of motility. And so to the extent that bacteria can think, it is thinking, okay, let me put the mineral here. Let me orient the cell, myself this way. And then I will start making the mineral out of this side. And that means I will go in this direction. And you know the direction it goes in? It goes in the direction towards oxygen. So I think it knows what it's doing, but it's just amazing that it can do all those things.
Yeah, and still on this, actually on this this physical aspect, this bacteria is also producing the SIC2 cytochrome that is oxidizing. Yeah, and is this protein localized near the? Have you seen any sort of localization of the protein on the membrane, or is it spread all over the cell?
Another rhetorical answer. Okay.
Sorry.
I wish I knew. So when we did the cryo TM, my hope was that we were going to see some sort of machinery here.
Okay.
And ultimately, my hope in making E. coli make Psych2 was to make enough to be able to make antibodies. We hardly made enough to be able to assay it, much less make antibodies. So, another dream dies. Of course, we could make antibodies to peptides. But I think the problem is, you can see there are not very many locations here, right?
Yeah.
With such low energy from each iron, it just doesn't make sense that there would be only this many areas of iron oxidation. So I think there's probably much more Psych2 here, and then there's some sort of shuttling mechanism to bring the iron to the nucleation site. So even more adaptation. What's interesting is that, I don't know if you can really see it, probably not here. The Gallionella stalk has more individual fibers. So, to each its own, right? To each its own structure.
Dania has a question.
Yes, hi, very great talk. I actually had a question following up on what you were saying about producing it in E. coli. What was your yield?
Oh gosh, I don't know offhand, but it was so low. We would grow liters and liters and liters and liters of E. coli. I think part of our problem was that only a fraction of it was folding correctly. And then also E. coli, I think it compromised the outer membrane of E. coli. And so E. coli was like a zombie in a sense. It was not really living and breathing correctly. And so we had to be quite careful about inducing the expression at the right time, because then at that point, E. coli would no longer grow. So that's not, I don't know the answer to your question, but it was barely enough to be able to do anything.
Okay, and I have a follow-up question. So have you, because I see that you have succeeded to express, it wasn't a recombinant expression, but to test the side hydroxidants.
Mm-hmm.
Have you also tried knocking out the gene and seeing if the dissolved iron reduction is reduced, or like if you have an effect of that?
That is the dream. We would love to do that. So we're actually funded to work with Jeff Gralnick at the University of Minnesota to make genetic systems. And, you know, it's very hard because for the most part, they don't make colonies. So standard genetics, where you screen colonies, doesn't work. Superoxidants under certain conditions might make tiny ones, but it's not very reliable. Okay, and so you have to do it in liquid culture by serial dilution, which is very hard. It makes everything slow. I told you everything was hard with these organisms. And so I think part of the problem also is that they're autotrophs that don't have very many other metabolisms, right? Superoxidants, yes. And so unfortunately, Jeff started with Mary profundus, and I thought, if you had asked me, that's not the one it's very beautiful, but it's not the one I would make the genetic system in, because it doesn't eat anything else. If you knock out Psych2, what is it going to do? Die? It's not good logic. So superoxidants, I think eventually we will try, but we have some other organisms in mind. Because Psych2 is very common, right? But we first have to show that the Psych2 is being used for iron oxidation in that organism, which actually, as it turns out, is not so easy. So this research is mostly an exercise in failure. But eventually we'll get there. You really have to love iron. And so we placate ourselves with these pretty pictures while we're failing most days.
Thank you and good luck.
Thank you.
So Feriel, you had a question too?
Yes, thank you very much. It was a very nice talk. I was wondering about, you mentioned some nucleation sites. So I wonder if they could have any other proteins just for biomineralization around. And if you were just, they must be expressed, perhaps their expression could be linked to cytochrome or things like that.
Yeah, so I mean, I'll just, for inspiration, put this up. This is all the genes, right? There are just so many genes. So the darkened boxes are the genes. There's so many genes we don't know what they do, right? And so it's really through concerted study of biomineralization that we are going to start to understand the other parts. As hard as the Psych2 work was, at least, it's a simple concept. It's the protein that makes the precursor. But then I think you're all asking the right question, then what happens? Right? Then what? Somehow it magically gets to the stock and everything happens? No. There's probably a protein that shuttles the iron. There's probably an entire system, a scaffold that moves that protein to the nucleation site. There's probably a protein that helps put it on the EPS. There's an entire EPS system, right? It's, I'm sure, very complicated. But if there's no model, right, we can't find that by homology if we don't know how it works. But surely in these various organisms, there must be some commonality. And so we looked at least, in concept to other systems that have genetic systems like the magnetotactic bacteria, right, et cetera, to try and understand. And we really hope that those systems yield general truths, and more specifically, general nucleation proteins that we can understand and find in our systems.
Have you seen any EPS around or things like that?
Yeah, so back to my PhD and postdoc work, we use synchrotron-based work to find that there are acidic polysaccharides within these stocks. And so they're not, it's a little bit hard to tell the original organic carbon from organic carbon that may have absorbed later. That's very tricky. That's why the synchrotron work is kind of hard that way. And it's hard in these particular organisms to isolate the organic, but actually that's why we're working with Leptothrix, because Leptothrix makes the sheath separately from the mineral. And so it turns out that that is actually very complex. It has both sugars and amino acids in it, and every single functional group you can think of. So I think we have bit off a different difficult problem, but it's tractable in other ways, including genetics.
Yeah, of course. Thank you very much.
Okay, do we have another question maybe? Okay, I think that's it. So, and it's right on time to conclude this seminar. So I wanted to thank you again really much for this really inspirational talk. It was really interesting. And I see you all next week for our next seminar that we will announce actually right now by email. So stay tuned.
Thank you very much.
Thank you again.
Bye.
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 Thank you. Bye.